Hi, my name is Ken Lasseson. I'm a data scientist, a software developer. I'm also a high-functioning autism spectrum person, and I have been interested in the microbiome for about 10 years and have developed a site looking at the microbiome. This is the first of a series of videos dealing with a series of posts which I put up looking at the state of knowledge which we have of the autism microbiome and looking at one specific child's autism microbiome results over 2 years, 12 samples. A very fortunate combination of information, and the parent has been very kindly giving me permission to share the results and analysis with everybody in hopes that it will help them. Okay, let's first take a look at a quick overview. Basically, I did a series of posts. You'll find those in the notes on the YouTube page. Technical study is basically kicking off what we know from published literature or from the site I've written using alternative statistical methodologies. I'm a statistician by training, and I often use techniques which medical professionals and researchers may not use for a variety of reasons, and sometimes those techniques get more interesting information or stronger information than the standard off-the-bookshelf methodology. So technical study on one looks at published science and citizen science and see, okay, where should we look for which bacteria? Bacteria is sometimes called taxa, T-A-X-A, or taxon, and we want to know which ones to look for. The typical microbiome sample has over 800 bacteria, sometimes 1,500. Identifying which ones are important, which ones are just noise, literally, is important, especially since diet and medicines will impact what shifts. So we end up with a very complicated environment, and we want to shortlist the bacteria which are most likely going to have an impact. The second post is dealing with applying information which we have gathered here to shortlist the bacteria and looking at one child's results. The second post is looking at something which is built onto the site I wrote, which is an artificial intelligence prediction of what some of the symptoms will be. This particular person, every single one of her 12 samples, number one prediction is autism. So by looking at that and looking at the combination of bacteria associated with autism, we ended up getting a short list of the bacteria which are most influential for this specific child's autism. We go on in the fourth post and we look at end products. Bacteria produces chemicals, metabolites. Those chemicals are feed into the body and into other bacteria. Some of them may cause inflammation, some of them may suppress inflammation. And we're looking at the bacteria associated with autism and the end products being produced and seeing if there are anything that sticks out. Is a particular chemical being produced by the bacteria very dominant? Why it's dominant is a different question, but we want to find out if we can identify what the chemicals being produced by bacteria are, which may be a key factor in the severity of symptoms of autism. And then we take that information and we apply it to this particular child. And we actually, I'll jump and tell you that we basically found that camel milk and awatere made a major change in the microbiome, but had a side effect that one particular bacteria just took off and climbing, so that this particular child has more of this bacteria than anybody else in 1,000 samples which is being uploaded. So that becomes an interesting question. Is this a good or bad bacteria to have? Or a correction, is that volume of this bacteria which this person have good or bad? My inclination on any extreme value is likely not good. but it may be a neutral. We don't know. That's one of the problems. We know so little about the microbiome that we are having to make educated guesses, and I'm trying to minimize those type of guesses. Okay, so that gives you the basic breakdown, and now I'm going to hop over to the first topic. In the first post at the bottom of it, you'll find the references, which goes and lists out all of the studies which I extracted information from. And as you can see, it's a reasonably good list. There's 20 studies. And what we do is we extract the bacteria shifts cited in the studies and drop it into a database so that we can take a quick look at it and say, okay, so what is the general pattern across all these studies? We also have included before this a copy of a summary from a review article done this year, which basically lists, none of the studies, most of them I've included, and what type of shifts are seen or done reported in that. So that is the same information, largely through a different accumulation method. Okay, now on my site you can find what the bacteria shifts are summarized in a cleaner format. When you go to the site, you see medical conditions listed there, just saying condition bacteria, which lists you all the things I've extracted information on, and it's a very long list, including things like hyperactivity, irritable bowel syndrome, etc. Often patients have multiple conditions, but let's assume we are dealing with autism pure. Then if you click on view bacteria for this condition, you will see the rank. Rank is a hierarchy. Genus belongs to family, family belongs to class, or other structurally like that. Then you find the name of it. You find the shift, is it high or is it low, and then you find the source of the study. So if you go down and look at something like Akkermansia, and you click source, it will give the source of the information, which is a 2019 studies with people in Ecuador. And if we go to the other one, we find that it is from a review which is cited from French Frontier Psychiatry, which reports the opposite results. Now, the interesting thing is we have 20 studies. We have extracted information from all of them. And what we find is 2 studies mention a shift was detected for Akkermansia. Just 2 studies. Another study indicated there's a high amount of this particular family. Just 1 out of 20 studies. Bifidobacterium, we have much more. We have 1, 2, 3, 4, 5, 6, 7 studies. But we have 6 saying low and 1 saying high. And what we have is we have inconsistencies between studies. And for autism, this type of inconsistency can be probably described to a couple of things. Number 1 is the diet of where the person is going to be a factor. 2, the healthy control versus the autism child. Both of them should be having the identical same diet. And unfortunately, what I've observed with autism is that often autistic children are put on special diets to try modifying symptoms. And now the question is, are these shifts results of autism or of the diet or medications they were put on to try to improve symptoms? If they were a case of either of the diet or the prescription, then they may not occur in native autism. They only occur in treated autism. And that's one of the problems with the studies. Now, a typical clinical study, you may well recruit autism from a particular locale and they come together. Unfortunately, probably half of the people in the study know each other from support groups. And in the support groups, they will share information. And the result is a high percentage of the people in that study with autistic children are probably trying to treat them in a very similar way, giving them the same vitamins, the same minerals, the same variety of things, which means that suddenly you have significance for that specific study, which isn't replicated anywhere else, because what you're actually measuring is not autism, but the shifts which were caused by shared treatment of autistic children, which makes things a little bit challenging to go somewhere. So this is the historical data. We can scroll down and you can see the number of times. If you want to know what the source of the information is, for example here, we click and again it hops over to the citation, and if you click here, you now get over to the article. Often the article which tends to be used more often than not are the full text article, which you can just connect to, and you can go through here and see all the technical description of it. You'll see things like Lactobacillus andoria, etc. being cited. In this case, we're also breaking down by GI symptoms in ASD and no GI system in ASD, which becomes a little bit more complicated in terms of trying to determine patterns. And so that's basically where we get the published studies from, which is from the studies. Now, the second thing we are doing is going over to citizen science. We have about 1,000+ samples of people with a whole variety of conditions, location, etc. And we also have 39 people who have uploaded who has autism as a diagnosis or symptoms. And what we can do is just go down here and click on this one, and now what you will find is, if you scroll down, you find autism official diagnosis. 27 here is being 27 bacteria or taxa which are being associated with it in some manner. And by just clicking on here, you will go over and see what the characteristics are. Now, you can do sorting here, so you can sort by class, by family, or whatever, by number of people who have this bacteria up to 39. For example, everybody has Blottia, and if you take a look at Blottia, we'll find that half of the people are in the lowest 25%. The lowest 25% is defined as a count between 0.19, which is almost nothing, and 5359, and then you can go and see the different levels as we go up in terms of size. So what we are doing is we're taking all the samples, finding the range across all the samples, and if the particular bacteria has no influence, we would expect the number in each one of these 4 boxes to be the same, somewhere around 9.75. That's not the case. What we find is that all of them are shifted to the low, so we have 19, almost half of them there, and then we have another 12, which is right next door to it. So low Blottia is something which, in our citizen science, using only the data which comes from people who have shared, we find that Blottia is a significant bacteria. And if you take a look a bit closer, most studies will publish if the significance is 0.002 or less. And as you can see, we are far, far more accurate. The statistical significance of just fine is far more than the majority of studies. And then we see other ones which are very common, and we can see the shifts and trends here. For example, bifidobacteria, for people reporting bifidobacteria, keyword for those reporting it, we find the majority of them are there. And what we will discover also is that there are some people who have practically none or who are on the low end. Small percentage, but it's there. Another one, which again we have high. So you can see that what we do is high and low. Here's another one where, although it's just percentage, you notice that, wait a minute, you have 6 out of 33 which are above the medium, and we have 27 which are below. Again, very significant find, and we now have a whole bunch of possible bacteria which are very likely associated with autism for the people in our sample. So, to flip over to the study, you'll find the links to both of those pages are sitting right there in a post. And what we have agreement between the published studies and our citizen science is we find agreement here for free with absolute clear, and then we have 2 where we have some agreement, as in some published studies agree and some do not agree. And then we have only disagreement on 1, which is this one is reported high in citizen science and low on published studies. The catch is, remember that often for bifidal we had something like 7 studies out of 20, only 35% of them reported any differences. So that is one of the problems. And then we also should be aware that age can be a factor. For bifidal bacterium and lactobacillus, the amount is much higher in children than in grown-ups, which means that unless we have enough data, which we don't at the moment, just to separate by age, some of this could be bias or confounders, because we happen to have a large number of children, so by definition we end up with a high number of bifidobacterium. And as these people become older, the bifidobacterium actually drops down to below average for adults. So again, there's complexity there. We don't have enough data. And then we go through, and what we also do is we expanded the list, seeing where we have things which published science at least twice reported the same thing, and there are no contrary studies. So we now end up with a greater list of bacteria which we want to investigate one by one for just one particular person. Now, why we are forced to do it for each individual person actually ends up coming back to DNA mutations. What we have here is from a study of DNA variations or mutations which results in a higher incidence of autism. We see there is a stack. Some of them are clustered. Some affect schizophrenia, others affect intellectual ability, others affect metabolic disorder, epilepsy, heart disease. So all of these could be in a child, may have just one of them, may have none of them. All of these simply indicate increased risk. The gotcha is DNA mutations also impact the microbiome, which means that some of the shifts which you see in the microbiome may be tied to particular mutations, which of course means trying to do a general one solution fits all runs into problems, because the solutions may well end up being specific solutions for specific mutations and specific microbiome shifts, and we don't have that volume of data available for analysis. Okay, so that's basically is the quick summary of the first post, which is reviewing the medical literature, extracting out the common bacteria reported, seeing where we are in agreement, and then from that agreement we could look at one individual microbiome and see what we can discover as to how well it matches a pattern or doesn't match a pattern. What we The goal was to identify the best candidates of bacteria of the 800 plus to look at. Have a list of 12 perhaps instead of 800, makes it a lot faster and more likely to find the right results that we can act upon. Okay, that's it. Stay tuned for part 2.